Okay. Granzyme-perforin pathway is an exotic pathway which is mainly used by the CTLs or cytotoxic T cells to induce cell death or apoptosis in the target. These are some of my disclosures. This slide compares the normal joints with the deformed joints in rheumatoid arthritis. The pathogenesis of rheumatoid arthritis is complex. As you have seen, many different types of cells, chemokines, complement systems, and cytokines, they are involved in the pathogenesis of rheumatoid arthritis. Some studies have shown that this particular disease originates in the temporomandibular joints. Other studies have shown there is a direct link between the chronic periodontal diseases and rheumatoid arthritis due to the presence of P. gingivalis bacteria. We are going to talk a little bit more about that. And more recent studies have linked the microbiome with rheumatoid arthritis and also lung-associated diseases with rheumatoid arthritis. Previous studies have shown that many mosquito-borne viruses can cause transient arthritis. This disease is diagnosed by the presence of anti-CCP or the ACPA antibodies and also rheumatoid factors. My major focus here is on the granzyme-perforin pathway-related genes. At-risk individuals or the early RA individuals develop autoantibodies approximately 3 to 5 years prior to the development of RA, but what actually drives the disease locally in the synovium is unknown. Synovium plays a crucial role in the pathology of many inflammatory diseases, including in RA. However, our knowledge of granzyme-perforin pathway during early RA and chronic RA is lacking. Early RA and chronic synovium has been classified into 3 different cell pathotypes: lymphoid, myeloid, and fibroid, but it is unknown what might be the drivers of these 3 pathotypes. Granzymes are xerin proteases. There are 5 different human granzymes, namely granzyme A, B, M, H, and K. Perforin is required to deliver granzyme into the target cells by CD8 T cells and NK cells. The effector stores of granzyme-perforin are complex with a proteoglycan known as serglycin. CD27 is highly expressed on the naive and activated CD8 T cells, CD4 T cells, and NK cells. Periodontal diseases has also been linked with rheumatoid arthritis. Here we will explore whether the level of granzyme-perforin pathway, related gene expression, and effector mechanism is related to the unique pathotypes observed and outcome in early RA and chronic RA patient in 2 different clinical trials, namely PEACE pathobiology of arthritis cohort, and the second one is stratified open level randomized phase 4 clinical trial. This slide simply shows that how granzymes are associated with the perforin and get poured into the cells by CD8 T cells into the target cell to induce apoptosis. And also there is a different mechanism. Some studies have shown that granzymes are also associated with the serglycin, which acts as a storage of these pro-inflammatory molecules and then again get into the cells through the perforin. There were 2 major goals of this study. In this clinical study, the first goal was to explore using the existing database by our collaborators to base related to bulk RNA sequencing and the extent of gene expression of various granzyme-perforin-serglycin genes present in the synovium and blood cell pathotypes of early RA and chronic RA. Chronic RA is also late RA. We examined the correlation between all granzyme-perforin-serglycin, -28 ESR, and -28 C-reactive protein. These are the 2 different clinical measures to measure the clinical disease score activity in clinic, and both are the ACR or the American College of Rheumatology approved criteria. We also examined the presence of various immune cells in the synovium from early RA patients using multiplex immunohistochemistry. Our second goal was to detect the presence of Porphyromonas gingivalis or antigen in the synovium from early RA patients. The synovial biopsies from early RA patients and also late RA patients or chronic RA were obtained by using an ultrasound-guided technology. In this case, we have obtained 6 to 8 synovial biopsies. The size of each biopsy was from 0.3 millimeter to 5 millimeter. Then half of the biopsies, they were fixed in the formalin for multiplex immunohistochemistry and histopathology, and then half of the biopsies were fixed in the RNA later buffer for 10x genomic analysis for bulk RNA sequencing. As I mentioned by our collaborators on this particular abstract, they have published in Cell Reports that early RA synovium and chronic RA synovium has been classified into 3 different pathotypes. Lymphoid pathotypes mainly have CD4 T cells, CD8 T cells, NK cells, and the B cells. On the other hand, myeloid pathotype, which is predominantly have the CD68 positive macrophages, both in the synovial lining as well as in the subsynovial lining area. And the last one is called autoimmune pathotype, or the fibroblastic or the fibroid pathotype, which have only the macrophages in the synovial lining area and they are absent in the subsynovial lining area. And most of the cells here are the fibroblasts. We compared the gene expression both locally in the synovium as well as in the periphery systemically in the blood, means PBMC. The top, all these box plots shows in the synovium and bottom one shows in the blood. It's clear from the data the lymphoid pathotype have the highest gene expression of all 5 granzymes and also perforin-serglycin as well as CD27, followed by the myeloid pathotypes. On the other hand, in blood, we do not see any difference in the expression or upregulation of these genes and perforin and serglycin and CD27 in the blood. So these data clearly shows that the expression is more local as compared to a systemic effect. In these studies, we have used 87 patients. These were paired samples. We not only have taken the synovial biopsies, but also the blood from the same patient at the same time. So we also compared then in chronic RA, late RA, the gene expression of all of these granzymes, and we found out still the lymphoid pathotype has the highest expression as compared to the myeloid pathotype. So these studies clearly shows that, and also the perforin as well as the serglycin and CD27. So these studies clearly shows that there is an upregulation in the lymphoid pathotype, and this expression is highly stable during the evolution of the disease from early RA to chronic RA. We also looked at the correlations between DAS28, which I mentioned is a measure of clinical disease score activity and is normally routinely used in the clinic. And also the correlation among all these 5 granzymes, including perforin, serglycin, and CD27. We found out the correlation specifically in the early RA synovium, they were not only positive but also statistically significant. On the other hand, we do not find these type of correlation in the blood. We also looked at the second measure, DAS28 C-reactive protein. C-reactive protein is a measure of marker of inflammation, mainly generated by the liver. And here we found out there was also positive correlation between DAS28, C-reactive protein, and all the enzymes in the early RA synovium. On the other hand, we do not see these type of correlation in the blood, although there were some correlations, but they were spotty and fragmented, and they were not even significant. So these data clearly shows that there is an upregulation of granzyme-perforin-related pathway genes from early RA to late RA, and also there are correlation during inflammation, particularly this pathway genes increases. Then we also looked at the absolute levels of C-reactive protein and erythrocyte sedimentation rate, because both are method of information. So here we found out that in late RA, we found out that there was perfect correlation, both very strong correlations, as you can see, between C-reactive protein and all these granzymes and the perforin. On the other hand, when we looked at the ESR, which is another measure of inflammation, we do not find these correlation, although there were some, but they were not statistically significant. So these data shows that C-reactive protein might be a better measure of inflammation as compared to ESR. We looked at the effect of two different drugs on the gene expression of this pathway. One is the rituximab, and second one is the tocilizumab. Rituximab basically depletes the B cell, and tocilizumab, which is a monoclonal antibody, blocks the IL-6 receptor. And in this case, we have taken biopsies from at week 0 and then after 16 weeks of treatment. Here we found out both of these drugs they decrease the expression of all these granzymes, including the perforin and the serglycin. Although tocilizumab that decreased much more as compared to the rituximab. Then the next question we asked, since we found that there is an upregulation of these granzyme-perforin pathway-related genes in the lymphoid pathotype, the next question we asked, what is the percentage of CD8, CD4 T cells at very early stage of the disease? Then we analyzed 23 early RA biopsies and using multiplex immunohistochemistry. And here we found out CD8 T cells were surprisingly, they were present at very early stage of the disease, and also CD4 T cells, and also CD8 and CD14 positive cells, which are macrophages. All these cells are also present. And this image shows the multiplex immunohistochemistry. We have used here a false color coding scheme to show all different types of cells. And then in multiplex immunohistochemistry, we could not examine the presence of whether CD8 T cells are present, whether these are CTLs or they are effector memory cells. So we examined the correlation between CD8A gene and among all these granzymes. CD8A is a gene which is specifically expressed on the CTL. And here we found out, interestingly, there was a perfect correlation between CD8A and all of these granzymes. And these data clearly shows that whatever the percentage of cells is present, CD8 T cells, this is very low, almost low, but these might be the CTL. And finally, we asked why CTLs are present, and we looked at the synovial biopsy. To do that, first we looked at the gum tissue from periodontal diseases, which normally have P. virus bacteria. Here we found the specks of these P. virus antigen in the gum tissue. On the other hand, when we examined the early synovial biopsies, the results were very surprising. Very rarely we found out in these early biopsies, we found out the specks of the antigens were present. And these were not actually rod-shaped bacteria, but actually these were the antigen fragments that were present. And the percentage of cells that have that on 4.5% as compared to 54% in the chronic periodontal gum tissue. Finally, the conclusion, the granzyme-perforin pathway upregulated in early RA and early RA in synovium but not in blood. The correlation between DAS28 ESR, DAS28 C-reactive protein and all granzyme in early RA confirmed the role of granzyme-perforin pathway in the evolution of this disease. Rituximab and tocilizumab treatment in R4RA affected granzyme-perforin pathway genes. The correlation between CD8A and granzyme confirmed that CTLs all are involved at very early stage of the disease. Minor specks of PG antigen in early RA synovium confirmed the direct involvement of this bacteria in the development of RA. CTLs might be targeting PG bacterially infected cells at very early stage of the disease. I would like to extend my sincere thanks to Dr. Michael Holders, who have provided tremendous support for these studies, and also to the faculty members, specifically Dr. Francisco Rosa. He has identified these first time the stacks of PG antigen in the early RAs, and also to many other investigators who have helped us to analyze the bulk RNA sequencing data and also provided the gum tissue from periodontal patients with diseases. Of course, we are amazed for the funding of this project for the last more than 25 years. Thank you very much.
So I asked you to compare that two major papers on complement activation. Have you compared the complement fragment in the granzyme expression profile? Are there any correlation or possible, you have your own data compared with your complement genomic data? Did you see any correlation with the complement? Fibroblasts also a very rich source to have a complement.
Yes, that's a very good question because there is a very hot topic. And last month, two papers came published in Nature that showing that granzyme basically cleave complement. And in these studies, we have actually, I'm a little bit stricter not to touch on that topic because that is done by our group, same group, so who are working on this because AMS is a study which is sponsored by the NIH. So I'm part of that AMS, so another group from Boston, they are working on that particular topic, and I'm working on a little bit different topic. As you see the last presentation, they are very closely showing also the CD8 T cell, they express granzyme K, right, and CD4 T cell express granzyme K. So I will not be surprising that granzyme K or other enzyme with M, H, and they also cleave complement and activate the complement locally. Thank you.
Thank you, everyone.